design and we can put the chemical inside the packet, or if you want that it should write within 2 days. So it can be, there are, by active packaging here basically we extend the shelf life by interaction of the package and the product and the internal environment. So that is an interesting development. Intelligent packaging. That is intelligent packaging. Even there are PTI indicators which can be designed and the status of the, to design the microbial status or package integrity or even free flowingness etc. Like a fruit here, we just example, let me have any fruit juice or fruit juice or vegetable juice, fruit juice. It is a good source of alcohol, that is the pH etc. But if although it is retarded in the thermal process, that is septically processed, but if due to any reason that processing or machinery has failed, technology has failed, so inside that fungi will survive and that fungi will grow and the ground your package you are not able to see what is there inside. You go and consume. So here using the nanotechnology, nanoscience, there is these sensors are designed, they can be put on the level that is a small microchip, it will indicate it will sign. So when you can design that this particular microorganism, its level goes beyond 10 to the power 6, 10 to the power 4, 10 to the power 5, it will glow red and that you discard it. Even have been developed by PhD, PhD students in my laboratory. Many of these technologies are patented. Some are already in transport industry.
Thank you, Commissioner, for a very they, of course, nutritional status, fortification, and the nutritional status of food, packaging, processing, everything which affects us day to day, right from what the kind of conversation we are already a bit late and scheduled, but still we could entertain 1 or 2 key questions.
This is a very good example, which Professor Gupta has presented to us, on how material science and advances in material science and understanding of material science can be effectively used and integrated into molecular biology. And this is something which is now becoming increasingly important. For example, recently, just about a few weeks ago, at the Tata Memorial Center in Mumbai, Professor Indranil Mitra, the very leading cancer surgeon and researcher, for the last about 5 years or 6 years, he's been doing a lot of work on chromatin fragments in the serum. And when such a thing happens, that when people normally earlier believed that chromatin, for example, chromatin fragments, because these cells are now dead, that people thought these are not very harmful. What he has shown very clearly through fantastic experimentation is that the chromatin actually goes into a lot of harm, because this chromatin has the DNA twined in it and it can go into the other cells and then actually pollute the other cells and create problems. So he has shown that if you remove chromatin from the serum and use the cleaner serum, that it gives you a lot of very useful effects. Now, to cut a long story short, what they have done then, collaborated with some groups where they use nanoparticles. And then they put the antibodies on that. Okay? And then how that goes and binds chromatin, and then that actually leads to reduction of the chromatin fragments. And there's a lot of work going on. So when you talk about nanomedicine, people are looking about into these type of methods of how to really exploit, and I use the word exploit, okay? Here you exploit these methods for the benefit of mankind. Professor Gupta, thank you very much. Thank you very much.
Actually, in the layman's language, I can say that
Mr. D.K. Gupta.
I think these oral competitive presentations. So can we invite Ms. Ankita Shukla Ankita Shukla from Department of Biotechnology, India, Gorakhpur University.
I am a research scholar from Department of Environment Science from Baba Saheb Bhimrao University, Lucknow. The topic formed through a biological process that causes the decomposition and degradation of organic material and converted into the hygienic humus waste product, that is compost. It's serving as a soilless media produced from diverse organic waste. Now, the compost extracts, this is also known as compost tea or compost watering extracts, are gaining increased attention as a crop protection tool for control of diseases and as an inoculant to enhance soil microflora. A large diversity of the 40 species of Fusarium oxysporum causes the vascular gland disease, affecting a great number of hosts, producing the serious economic loss. These pathogens can exist for a long time in absence of a suspectable crop due to the production of the spores. The stimulation of the population of antagonistic microbes in the soil amended with various organic compost was also shown to reduce the survival of Fusarium pest pathogens. Now the question is why we use compost. They are mainly the resistance variety we can use, but these resistant varieties can provide some degree of control, not completely, and have a high cost. If you use the chemical agent, they have high frequency and cost. They also affect the non-target organisms and they develop the resistance in pathogens. And due to the continuous use of these chemical agents, and they also cause the risks to the human health and also surrounding environment. The importance of compost in the agricultural field, because when the compost mixes soil, it's degraded. These compost changes into the organic complex in the compost changes into the inorganic substance, and they act as a nutrient supplement in the soil. By adding the nutrient supplement, they improve the quality of soil and enhance the plant growth. And also they suppress the soil-borne pathogens. And because it is a part of solid waste, by using as a compost in the soil, it helps in the solid waste management practices. And by this, it helps in reducing the environmental contamination. Maybe I have selected two pathogens for study. First is the Fusarium oxysporum F species cycling that causes the chickpea wilt. Chickpea is one of the most important pulse crop grown throughout the world. It is the third important food legume after the bean and pea. The next pathogen is the Fusarium udum that causes the most serious and destructive disease of chicken pea. Chicken pea is the most important pulse crop grown in UP. I've taken the sample, I've taken the pathogens isolated from chickpea and chicken pea fields from 30 village near LCPGI Lucknow. And second is the poultry manure, that is the chicken manure. 4 types of compost treatment which are taken. First is the treatment, which is by mixing the wheat straw and cattle manure in 1:1 ratio. Second treatment is by mixing the rice straw and cattle manure in 1:1 ratio. Treatment 3 is only the taking the cattle manure, and T4 is only the poultry manure. All the compost extract is prepared by mixing the compost with autoclave phosphate buffer in 1:2 ratio and shaking for 10 minutes. Oxygen centrifugated at 5000 rpm for 10 minutes. The residue is discarded and supernatant work as a active compost extract. I similar in vitro 0.5 gram of autoclave and unautoclave compost in different concentration of 5, that is 5, 10 and 15% of compost extract that is prepared in a phosphate buffer. Each one added with sterilized PTA medium, that is Rebexox agar, and poured in the bacteria. After solidification of the medium, 5 meter diameter disc from 7-day old culture of both Fusarium species was placed on the center of each disc separately and incubated for a week at 25 degrees centigrade. Fusarium udum, the same results were shown that the unsterilized compost has much inhibition potential against these both pathogens. compost reduce the growth of both the pathogens while the autoclave compost has breeds effect. The T1 and T2 causes the maximum suppression in unautoclave conditions. The concentration of T1 and T2 treatment of compost extract causes the maximum inhibition. Note that the compost extract have high suppressive capacity against both the pathogens in comparison to direct addition of compost. Compost extract prepared from combination of plant waste and animal manure have effective potential against both the pathogens. The maximum suppressive capacity was observed by extract prepared from combined mixture of cattle manure and leaf straw.
End-to-end producer. So it is obvious that you have taken some other species. May vary, particularly Jatropha conjurencies. So that vary. So this is the interspecific hybrid and fruit production is very small. So probably may vary. 8,39,052 insect species reported from the entire world. 21,000 species, that is 6.08% are reported in India. These are some biodiversity. These are the commercially exploited species of the silkworms in India. This bombing sword is mulberry silkworm, Anthera america is tussar, and Anthera pralea is oak tussar, and Samia richly is the hairy silkworm, Andy, and Anthera asama is Muga silk.
On the two polymer system, known as the polymer and the polymer. That is for the transfer delivery system. So why I have used this polymer? This polymer has especially the polyanisopropilenite is a very unique polymer. When it remains at a room temperature, it's a transparent liquid. But as the temperature goes up around the body temperature, immediately it converts to gel state. So what happens? It can trap the molecules which are inside the liquid state. So what happens? It's soluble in the water. Because of it forms the hydrogen bond with the water molecule. Once as temperature goes up, above the LCST means lower critical solution temperature, immediately the hydrogen bonds with the water start weaken and the molecule gets thickened and it forms the gel state. So polyvinyl alcohol, boil it and make a film of polyvinyl alcohol. We have carried out 3 different experiments. One with the PVA film, another with the PVA film at 20 degrees Celsius, and one with the PVA American film at 40 degrees Celsius. So we have checked the release pattern at 2 different temperatures, one at 20 degrees Celsius, another at 40 degrees Celsius. So what are the changes we have found in how the release pattern gets changed when the temperature goes up? So I would like to show that results. So first this graph I will show that this is the emission spectra, fluorescence spectrophotometer. We have taken a fluorescence spectrophotometer. Spectra shows that these films do not have any fluorescence properties. They are without fluorescence property. And the other one, we have taken the full spectrum scan starting from 200 nanometer to around 700 nanometer, and we got that the lambda max of the absorption maximum of the ciprofloxacin is around 274 nanometer. This lambda max was characteristic for our work, and this was the emission spectrum of PVA film loaded with the ciprofloxacin. What we did, we have taken the ciprofloxacin around 50 microliter, and then we have loaded it, mixed it with the methyl alcohol water mixture, and we have kept our film inside that liquid system. And the first one with the PVA film, second one with the PVA minipan film. So the approach is like that we have PVA is not having any trapping property to any metal molecule, but this minipan has the property to trap any molecule, drug molecule or whatever it is. So we have prepared the film of these 2 polymers without any cross-linking. We haven't used any cross-linker here. Simply we have taken the solution of PVA, taken the solution of minipan, mixed both of them, dried it for 48 hours, and then we have cut it over a quart strip, which is having size around 1 centimeter, then 1 centimeter, 1 centimeter to 4 centimeter. The same thing with the happens with the PVA minipan. No, it's with the 40 degrees Celsius. With 40 degrees Celsius, we can see that to release to achieve the OD of around 0.11, the drug has taken around 8 hours. It has taken around 8 hours. But in the case of at 20 degrees Celsius, things remain same. Once again, the pericarp cannot trap any drug molecule. It's immediately at low temperature continuously immediately release the drug. But in the case of PDA pericarp film, which are at 40 degrees Celsius, the pericarp goes as temperature rises, it renders, it traps the drug and it inhibits the release of drug. So this is what our approach. So we have found this is the document slide of at 40 degrees Celsius. This study of the release pattern of the drugs of PDA pericarp at 20 degrees Celsius. And my guide, Dr. A.K. Mishra, Dr. S.S. Ray, and Dr. Manish.
My topic is study of physiological and biophysical alterations in a diasymptotic sonography under organic stress. I had done this work under Dr. Shanti Sundaram, Center for Biotechnology, University of Karnataka. Background of my work. Main point is pollution. Due to enormous quantities of organic and inorganic compounds of polluted compounds are released into the environment due to human activities. Which are the pollutants from the industry. Phytic compounds can be degraded by cyanobacteria. That's what I want to tell you, because cyanobacteria are photoautotropic. They can synthesize their own food, because normally Pseudomonas bacteria is used for this work. Phosphorium, which is a filamentous cyanobacteria grown in BD11 medium, given by Steiner et al. Pseudococcus, which is a unicellular coccal type of cyanobacteria grown in same BD11 medium and supplied with nitrate source. Third one is Spirulina platensis. About Spirulina, you all know Spirulina can be edible. Cells are used. These are grown in sterilized medium with sterilized medium under 27 degrees Celsius. Growth and survival and growth were measured. Phytic compounds and pyrethroquinone, which I have supplied, is 5 millimolar concentration to 400 millimolar concentration I have used. This, but up to 200 micromolar concentration, there was no change in cell death, cell growth. Carotenoids and phycocyanin, because cyanobacteria is cyanobacteria is also coming from phycocyanin. Cell and spiroplast, which are spiroplast isolated and whole cell, both we have checked. Here are the results. Growth behavior up to 7th day, because it was very low rate of growth. Maybe the cells are trying to assimilate, or the all proteins were synthesized slowly in the esophagus, all maybe helping the cells to survive. They have studied benzene to agree sardine and PNP. The 0 day, 24th hour, 48th hour and 7th day. I have checked all chlorophyll because 24-day data is too much, so I have given only 7 days. Sorry, carotenoids. I have to say carotenoids also have antioxidant properties. So carotenoids are the